Hello everyone, I welcome you in Revise with Me episode. In this video, I will discuss all the things about plasma membrane I have read so far. Plasma membrane is dynamic and fluid structure which form outer boundary of cells. Most importantly, it acts as selectively permeable membrane and regulates molecular traffic across the boundary. Then different models were proposed to describe its structure. The most accepted one is fluid mosaic model. Well, it was given by J. Sanger and G. Nicholson in 1972. In this model, membranes are viewed as quasi-fluid structures in which proteins are inserted into lipid bilayers. This model explains actually two things. One, mixed arrangement of proteins surrounded by lipids, and second, fluid movement of lipids as well as proteins alike. Here you can see plasma membrane forms bilayer structure in which both proteins and lipids move freely, but their movement of either from one phase to another is restricted. Now, chemical composition of plasma membrane. Plasma membrane contains proteins, lipids and 5 to 10% of carbohydrates. Carbohydrates when bind to lipids it form glycolipids, and when carbohydrates bind to proteins it form glycoproteins. Carbohydrates are sufficient in the plasma membrane of eukaryotic cells. Now let's move towards lipid bilayer, which is the basic structure of plasma membrane. This bilayer is composed of two leaflets of amphipathic lipid molecules. Amphipathic means having both hydrophilic as well as hydrophobic side. Here head is hydrophilic, or we can say polar, and is in contact with extracellular aqueous phase, and two fatty acid tails which are hydrophobic in nature. Then there are three classes of lipid molecules present in lipid bilayer: phospholipids, glycolipids and sterols. Phospholipids. In phospholipids, head is hydrophilic and two tails are hydrophobic. Actually, phospholipids are made up of four components: an alcohol, it may be glycerol or sphingosine, fatty acids, a phosphate, and that phosphate is attached to an alcohol. And two types of phospholipids are there. One is glycerophospholipid. It means when phospholipids are derived from glycerol. It's also called as phosphoglycerides. So it contains glycerol, fatty acid, phosphate and alcohol. For example, choline. Then second type is sphingophospholipid. They contain amino alcohol called sphingosine instead of glycerol, and a fatty acid, phosphate and an alcohol attached to that phosphate. Sphingosine is linked to fatty acid by an amide bond. Sphingomyelin is the most abundant sphingophospholipid. And plasma membrane of animal cells contain four major phospholipids: phosphatidylcholine, phosphatidylserine, phosphatidylethanoamine, and sphingomyelin. Then glycolipids. They are either monosaccharide or oligosaccharide and are covalently attached to lipids. Simplest glycolipid is cerebroside. It only contains one sugar residue. It may be either glucose or galactose. Gangliosides are most complex glycolipids. The glycolipids are found extensively in outer leaflet of the plasma membrane with their carbohydrate portion exposed to the cell surface. Then comes sterols. Here we have learned steroid nucleus is basic structure and it contains 4 fused rings, 3 with 6 carbons and 1 with 5 carbon. It's planar and rigid. Cholesterol is the major sterol present in plasma membrane of animal cells, but plant cells lack cholesterol. Bacterial cells also lack cholesterol. Then comes lipid arrangements in bilayer. Here lipids are not randomly mixed with each other. Cholesterols are organized in aggregates called the lipid rafts. They are microdominous, heterogeneous and contain specific proteins, and lipid rafts are of two types, planar and caveoli. Caveoli are flask shaped and covers the plasma membrane, and it contains caveolin proteins. Caveolin is an intrinsic membrane protein and is a cholesterol binding protein. After that there comes amphipathic lipids. When they are mixed with water, three types of lipid aggregates can form. Okay, so this was all about plasma membrane.